Welcome. In this session, I'm going to discuss about viruses, viroids, prions, and lichens. So, in the old textbook, you don't find information on prions. In the new textbook, they have introduced a small paragraph after viroids. Okay, so let's begin this session with virus. So, here the meaning of virus is venom or poison. So the meaning of virus is venom or poison, and this term virus was first coined by D.J. Ivanovsky in 1892. So he is the first person to give the term virus, and here the viruses are obligate parasites. Now, what we mean by obligate parasites? Obligate parasites are the parasites that live only as parasites. If host is not available, then they cannot survive. They can live as parasites only if the host is available. Of course, there is a big classification of parasites. We see facultative parasites, and I'm just telling the word opposite to this obligate parasites. So there are facultative parasites. Okay, so in case of these facultative parasites, they will live as parasite if host is available. If host is not available, they live freely. Okay, but they don't die. But in this case, obligate parasites, if host is available, they will live. If host is not available, they cannot survive outside the host. So all those parasites who exhibit such phenomenon, they are called obligate parasites. And okay, and they are inert outside the outside the host cell. So here, which means here inert, it doesn't show any living signs. Okay, so it doesn't show any living activity. So the meaning of inert is that it doesn't show any living activity outside the host cell. Okay, so they are obligate parasites and inert outside the host cell. Then the viruses are not truly living. So that's why we did not classify under any of the kingdoms, right? Even I told you, after 5 kingdom classification, 6 kingdom classification has come up, and even 7 kingdom classification have come up. Okay, and going to even get an 8 kingdom classification, and none of these classifications have included these viruses. Okay, so there is a controversy, okay, that whether to consider them living or non-living. At present, we are considering them as non-living only. Okay, right? And some authors, they say that they are connecting link between non-living and living. So at present, we are not considering anything the living. Okay, and we did not include it in any of the classification. So viruses are not true living, and they are acellular, and they are acellular okay structures, means they don't have any cellular organization, so they are acellular structures. Then, so viruses have crystalline nature. So means when they are present outside the host cell, we can make them into crystals. So we can make actually non-living, certain non-living things, we can make them into crystals, not the living ones. So here this is a non-living property. So viruses can be crystallized. So that is a non-living property, and it is not a living property because no living organism is capable, okay, or can we make into crystals, but viruses we can make into crystals. So that's why, so they have, okay, a crystalline, they are crystalline in nature. Okay, so outside the host cell we can make them crystals. And I'll put it in another words like crystalline inert crystalline structure. So inert crystalline structure. Then next one, the viruses are smaller than bacteria. So viruses are smaller than bacteria. Okay, then the viruses actually infect the cell and they will take over the control of the cell, and they use that cell for the purpose of replication, and during this process they will kill the cell. Then in 1898, okay, so that's in 1898. So here in 1898, a person named M.W. Bejernik. So a person named M.W. Okay, Bejernik. So he conducted an experiment. So he conducted an experiment, and in that experiment, actually, he shown that extract from infected tobacco plants. So what he has taken is, so from infected tobacco plants, okay, he has taken an extract. So that's a fluid. Okay, so he has obtained a fluid from this infected tobacco plants. Okay, so it's called the extract, and he applied this extract to healthy tobacco plants. So to healthy tobacco plants, and when he applied this extract that has been taken from infected tobacco plants to healthy tobacco plants, what he found is these healthy tobacco plants have shown the signs of the infected tobacco plant. So whatever that symptoms that were shown by these infected tobacco plants are also seen in healthy tobacco plants, just when he applied the extract taken from infected tobacco plants, right? So extract, of course, this is a fluid, so hence he called contagium vivum fluidum. So he called this fluid as contagium vivum fluidum. So here contagium, which means infectious, right, and vivum, which means, okay, living, which means living, and fluidum, which means fluid. Okay, so when translated to English, it's called infectious living fluid. It's called infectious living fluid. Okay, and I already told you that these viruses are smaller than bacteria. So how we come to know that they are smaller than bacteria? Because they pass it through bacterial proof filters. So when they used even the bacteria proof filters, it passed it through those okay, bacterial proof filters. Okay, and because of that we came to know they are smaller in size when compared to bacteria. The next one, and another person, W. M. Stanley, in 1935. So W. M. Stanley in 1935. So this person, okay, showed that the viruses can be crystallized. So he showed viruses can be crystallized. So crystallization is a property of non-living things, right? So he showed that viruses can be crystallized, and these crystals, they mostly consist of proteins. So whatever that crystals he made, so those crystals consist of mostly proteins, means they are made up of mostly proteins. Okay, so then, okay, so that's an information and introduction on viruses. Now let us see its structure. So here viruses are made up of two components. So they have two components. One, so they have a protein coat. Okay, so this protein coat will be protective in function, and then it has a nucleic acid. Nucleic acid. Okay, a protein coat and nucleic acid are the only two components of a virus. No cytoplasm, okay, no cell organelles, nothing. So there is no cellular organization present at all. So just a protein and a nucleic acid is present, and this nucleic acid can be either RNA or DNA, but not both. Okay, we know from Monida to kingdom Animalia, all of them will have both the nucleic acids. So they will have RNA as well as DNA. Of course, only DNA will act as its genetic material, whereas the RNA acts as non-genetic material. But in these viruses, the viruses will have, okay, either RNA or DNA. In single virus, you don't find both DNA and RNA. So for that reason, some authors also have classified viruses into two categories. Okay, DNA viruses and RNA viruses, which means the DNA viruses are the one who have nucleic acid DNA, and RNA viruses are the one who have nucleic acid as RNA, right? So those are the two components, that is protein coat and nucleic acids present here, right? The next one, I mean here, because of this, I can also call it as a nucleoprotein. Also call it nucleoprotein. Okay, so a virus is a nucleoprotein because it consists of a nucleic acid and a protein. Next, okay, in general, the viruses infecting plants. So in general, so viruses infecting plants. I told you already, viruses are obligate parasites. So all of them are parasites. There is no exception in this case. So in general means the viruses that cause diseases in plants. So in general, viruses infecting plants. So they have a single stranded RNA as its genetic material. Okay, so this nucleic acid is the genetic material, so that is RNA or DNA is considered the genetic material. So is the one that will be, okay, passed from one generation to another generation. So like from parents to offspring, and those offspring becomes adult, and from them again to their offspring. So that material, we actually call it a genetic material, and it contains all the information, okay, for the characteristic features of that particular organism. Okay, so that is the genetic material. So here the viruses that infect plants will have single stranded RNA molecules. Okay, so usually RNAs are single stranded. So here SS stands for single stranded. So that single stranded will be looking something like this. Okay, so I'm just giving an example. So this is how it looks like. So these are all the nitrogen bases. So later we will study about them. Okay, so the G stands for guanine, cytosine, A for adenine, U for uracil. Okay, so if we see U in the case, then it will be RNA, and if it is replaced with T, then that will be DNA. Right, so those things we will discuss in detail in the chapter molecular basis of inheritance. So just I'm giving, okay, a basic idea. So single stranded RNA looks somewhat like this. Then coming to the viruses infecting, okay, animals. So in general, or usually, viruses infecting animals. So in this case means all those viruses that infect and cause diseases in animals, they may have, okay, single stranded RNA, or they may have double stranded RNA, or may have double stranded DNA. Okay, so this here which I have shown is a single stranded RNA. So if it is a double stranded RNA, then we find two strands in this case. I'll take the same basis, okay, A U, right, and there will be pairing. Okay, the pairing we'll see it later. Right, so this is how we see a double stranded RNA. So this is a double stranded RNA, and this one is a single stranded RNA. So in general, the viruses that infect animals may have single stranded, so like this, or may have double stranded RNA, or the double stranded DNA. So the double stranded DNA will have, okay, something like this. Okay, so here I just gave, so this is a double stranded DNA molecule, how it looks like. Okay, so in detail, I told you that we will study it later. So the next one, okay, viruses infecting bacteria. So viruses infecting bacteria. So the viruses infecting bacteria are called bacteriophages. They are called bacteriophages. So viruses infecting bacteria are called bacteriophages. Here actually viruses, we can also call them simply, okay, phages. Okay, not phases. It is, okay, phages, Phages, right? So viruses or phages are one and the same. And here bacteriophages are not bacteria. The bacteriophages are actually bacteria, I'm sorry, are actually viruses that infect viruses. So bacteriophages are viruses that infect bacteria. So viruses that infect bacteria. So in these viruses, we usually find double-stranded DNA. So its nucleic acid will be usually double-stranded DNA. right? So now let's see the structure of a tobacco mosaic virus. Tobacco mosaic virus. So in short, it is called TMV, and this causes a disease called mosaic disease of tobacco. So it causes a mosaic disease in tobacco plants. So that one is actually tobacco mosaic virus TMV. Now coming to its structure, I told you that a virus is covered by, or it's made up of a protein, and this protein coat is called capsid. The protein coat is called capsid. So here we shall see. So this is how the protein coat in case of the tobacco mosaic virus looks like. So here this protein coat has many small subunits. So that is, capsid has many smaller subunits. So those smaller subunits are called capsomeres. So those subunits are called capsomeres. So I'll show here what these capsomeres are. And so here we have the nucleic acid. So in this case, the nucleic acid is RNA. So this is RNA. Of course, this is single-stranded RNA. So tobacco mosaic virus. So this is TMV. The tobacco mosaic virus have single-stranded RNA. So this one is the protein code. So whatever we see is the protein code, and these individual units we see here are capsomeres. So those individual units are called capsomeres, and these capsomeres are arranged in helical or polyhedral geometric forms. Okay, helical or polyhedral geometric form. So in this TMV, actually the capsomeres are arranged in helical shape. So here it goes somewhat like this. So they are arranged in helical shape, right? So that in TMV, the capsomeres are arranged in helical geometric form. In helical geometric form. Okay, so this is about the tobacco mosaic virus. And here one more point, this tobacco mosaic virus is the first discovered virus. So it is the first discovered virus. And now we'll see one more virus. So that is bacteriophage. We'll see the structure of bacteriophage. So already I told you bacteriophage is actually a virus that infects bacteria. So that infects bacteria. So here in this bacteriophage, the protein coat, that is capsid, have capsomeres, and they are arranged in a polyhedral form. So arranged in, okay, polyhedral form. Okay, so we can see here so they are arranged in polyhedral form. So this part of the bacteriophage is called head, and inside this head it consists of its nucleic acid. So if you open it, inside you find the nucleic acid. So head encloses nucleic acid, and we know the type of nucleic acid in bacteriophage, it will be the double stranded DNA. So it is double stranded DNA, right? And below this we find again protein. So all these are made up of, okay, proteins. Okay, so this will be a collar, and below the collar we find a sheath. So this is arranged like this. Okay, so all this is made up of protein only, right? So this part is the sheath, and here we have the collar, and at the end it has a basal plate. So it has a basal plate. So this is basal plate. Okay, and this one supports the tail fibers. So it supports the tail fibers. So here these are the tail fibers. Okay, so I'm just drawing. So these are tail fibers. Okay, and here we see the basal plate have some structures, and these are called spikes, okay. So actually this one, once it sits on a bacterium, say this is the bacterium, and okay, so it sits on bacterium like this, so with its spikes, right? And inside this is the nucleic acid, so that is DNA. So now this sheath actually can contract. As a result, what happens is, okay, so this portion can come very close. You can see here it has come very close, contracted, and then okay, it will help in injecting that nucleic acid into the bacterial cell. So this is a bacterial cell, and this is, okay, the nucleic acid DNA. Okay, so during infection, actually, it will actually send the nucleic acid which is present in the head into the bacterial cell, right? So this is the structure of a bacteriophage, right? Next, viruses cause several diseases in humans, okay, in animals, and so we'll see here in humans, okay, so different diseases caused by viruses are like mumps, right? And then so they also cause smallpox, and then herpes, okay, then influenza, AIDS, etc. So these are all different diseases, okay, caused by viruses in humans. So we see mostly in detail in a chapter called Human Health and Diseases. So just I'm mentioning, so there we will see what mumps are, okay, caused, okay, and the name of that virus: smallpox, herpes, influenza, and AIDS. So here especially these herpes and AIDS are STDs, so that are transmitted by, okay, sexual contact. So here herpes is a STD, so that stands for sexual transmitted disease. So AIDS is also a STD, sexual transmitted disease. Then coming to plants. So in plants, okay, viral infection shows different symptoms. So the viral infection shows different symptoms. So what are all those symptoms? We will see, okay, there will be mosaic formation, means on the leaf you will see patches of white color, so where actually the leaf will be green in color due to loss of that, okay, chlorophyll, or the cells they die, okay, especially the cells will die in those locations. Because of that, you will see patches on the leaf, and that we call a mosaic formation. We see the mosaic formation, then we will also see the leaf rolling and curling. Okay, so the leaf will be, okay, rolled, or it will be curled, so it will show a lot of curling. Okay, so because of that it may not be able to get enough sunlight, and so even if rolling also takes place, the leaf will not be able to perform proper photosynthesis. Okay, hence the productivity of that plant decreases. Then alloying, okay, and vein clearing is also seen. So here those parts, some of the parts may turn into yellow color, especially the veins where these certain cells they die. So what happens is the vein will turn into, okay, yellow color, so that will be seen very clearly. So the remaining part will be, okay, green in color, and the veins will be yellow in color. So that's, we call it okay, yellowing, okay, and vein clearing. Okay, so here these veins will be seen all in yellow color. So it's a vein clearing we see in that condition. And then, okay, dwarfing means the plant will not grow properly. Okay, and then uh, stunted growth. Okay, stunted growth means it will not grow, okay, properly. So here dwarfing means it will stop growing, as a result the plant remains short. So that is in case of dwarfing. And stunted, okay, in case of stunted growth, the plant grows sometimes stops, okay, it will not show a proper growth, right? And you can search Google, right, so for mosaic formation, how the leaf looks like, then leaf rolling, how it looks like, the curling and yellowing. Okay, so the lamina may turn into yellow color. Okay, so of course this condition we call it chlorosis. So chlorosis is changing from green color to any other color, is actually called the chlorosis. And here in vein clearing, what happens is the vein, and or I'll simply say chlorosis of vein and veinlets. Okay, chlorosis of vein and veinlets. You can see them, okay, in Google. So all those pictures about, if you do not follow how the mosaic formation is seen, the leaf rolling and curling, yellowing and vein clearing, and so. Okay, with this I will end today's session. So thought of discussing the remaining viroids, prions and lichens. That part we will see in the next session.